Mitochondria altering the membrane potential, and you'll hear a lot about that in the next few minutes. PINK1 doesn't get cleaved. So if we go back, that's a PINK1, and this is a cleaved version of PINK1, and that's what happens. That's what should happen when the mitochondria is being damaged. You get a decreased amount of cleavage and an increase in the non-cleaved portion of PINK1, which in turn recruits a protein called FBXO7, a pretty rare Parkinsonian gene, which in turn recruits Parkin, one of the first Parkinson's genes. So you've got 3 Parkinson's genes, and the evidence for this I haven't got time to go into. It's really quite strong, all pulling together in one pathway, which in turn regulates the control of the mitochondria and whether we turn it over. And so you can imagine that mitochondrial biology has become rather central in our thinking about Parkinson's disease in the last 5 or 6 years. Okay, so using the human as an animal model, we started, a few years ago when Andre first came here with just a few interactors. It's not quite as complex as this yet, but was building towards this with a more complex view of the pathways involved in Parkinson's disease. And we're starting to get hold of patients using those as an animal model that can build cohorts that we can start to validate and discover biomarkers based either on the biology and something from samples from patients, and then in time we're building what's called the Wolfson Experimental Neurology Centre, £20 million award from the Leonard Wolfson Foundation to build a facility in the hospital just down the way there where we can take patients who are at risk of Parkinson's, Alzheimer's, motor neuron disease and neurodegenerative conditions and use them for first-in-human studies. So the first time we have a chemical that we think might improve Parkinson's disease, we are going to have a facility to do those experiments in the human.
You said people at risk had, very important, how do you know they're at risk?
Well, that's a good question. So what we are going to use is at risk, we're going to use genetic profiling as the at risk. Not because it's the only one or even the commonest, but because it's a solid place to start with.
Yeah, okay.
So we will start with people who've got a PINK1 mutation or alpha-synuclein mutation, which are pretty damn rare. But if we then can discover things about Parkinson's, we then will ask the question, okay, in that form of Parkinson's, does it now work for, you might call the common or garden variety of thing? Now, whether that link happens, we'll have to see.
The genes that we're talking about, the expression of that gene into a Parkinsonian disease phenotype is always 100%, so are they deterministic, like the particular mutation?
No, they're not 100%. Some are very high. Sometimes it's 60-70%. So they're pretty high from our understanding,
okay,
but they're not 100%.
So meaning if you can select people with that gene mutation, that's relatively good controls group, yeah, to see whether you can have any preventive measures. Or
I mean, if we had endless amounts of money, and we as a country had endless amounts of money, you probably wouldn't need to start with it. What you'd start with is just go out to these very large cohorts of people. So, for example, they've been following a cohort since 1946, and it's called the 1946 cohort. And there's a bunch of people who are born within 1 week of 1946, and we've been following them since then, every year, every few years, taking blood pressure, serum this, serum that, and all that sort of stuff. They would be a great cohort to see which of those go on to get Parkinson's disease or Alzheimer's things. But even though the cohort's quite large, it's several thousand, but I can't remember. Let's say it's 10,000. I can't remember the number. The frequency of Parkinson's isn't still the numbers are going to be rather small, and their causes are going to be numerous. And so even if you have a very large cohort, even with a common-ish disease like Parkinson's, you still don't have the power to make the observations you need. So that's why David Cameron announced just before Christmas that they're going to put £120 million into finding rare diseases. And thankfully, he includes familial forms of Parkinson's disease as rare disease because it meets the criteria. So we hope, because he recognises, and the drug companies recognise, that this is a simpler, although complex, a simpler route into drug discovery. I think I'll stop there because I'm talking enough. Any questions so far?
I would have
industry in this at all?
Yes, so you mean sort of drug company type pharma, yeah. So I just came from a teleconference I had just 15 minutes ago with somebody from GSK. They are interested in Parkinson's disease, as many of you will be taking some GSK medications, I presume. And they're interested at the moment in LRRK2, and we were talking about LRRK2, and they're taking the view of people who carry LRRK2 mutations and doing things like scans and ultimately spinal fluids and urines and all sorts of things. So drug companies are getting interested in it because they've worked out that the model that they've been adhering to for the last few decades of common disease, diabetes, Alzheimer's, Parkinson's, hasn't really produced very many drugs. Not for neurodegenerative conditions. It's been a real desert, really. I mean, you think
money in this one.
well, no, yeah, they can. They can also see it's slightly more permissive framework for regulation. I'm not a drug regulatory expert, so I might be overstating here, but my understanding is that it's a bit easier to get a drug to market for a rare disease because it's seen as being a bit special case, and so the passage through the regulations is a bit easier. I've decided that if they can find drugs that work for, I don't know, a few thousand, it's still better than having a failure that could have worked for tens of thousands.
Can I have one more question?
Sure.
Is there a research paradigm or like avenue where the idea would be to have like a risk, like a group of people at risk, and somehow do some research on finding commonalities in those cases? Of course, it would be a longitudinal follow-up study. In those cases where you don't find the occurrence of the disease.
For I think if we understand the genetic architecture, we will define people with, let's say, some increase in risk. Sometimes it might be 100% because of some genetic mutation. Sometimes it might be a rather moderate increase in risk because they've got a range of genetic modifiers of disease risk, and we'll follow that group and predict that they're more likely to get into Parkinson's disease and try and find signals or exposures to chemicals or whatever it is that's turned them into the disease.
Yeah.
But that is, as you say, longitudinal and long. That's a hard graph.
And of course very hard because it can be like completely open-ended lifestyle choices even that can have an effect. So.
But I think that's the best chance of us getting somewhere in the gene environment risk factor.
Thank you.